Welcome to Diagnosing Dx, the podcast giving diagnostics the platform it deserves. While therapeutics and devices often take centre stage, diagnostics is the engine driving precision medicine. I'm your host, David Philby, and each week we spotlight the innovators, breakthroughs, and real-world challenges shaping the future of DX. This episode is about how we actually move biology forward. I sat down with Serge Saxonov, founder and CEO of 10x Genomics, to talk about the launch of Atera, a new platform that enables whole transcriptome analysis in tissue at scale with single-cell level sensitivity. We get into why biology has been fundamentally limited by the way we measure it, why most technologies will never make it into the clinic, and what changes when those constraints are removed. We also go deeper into what it takes to build a category-defining company in life sciences, from talent to culture to maintaining innovation as you scale. Welcome to the show this week. How are you?
I'm great. Thanks for having me.
Absolutely. Where in the world are you?
I'm in Pleasanton, California. That's where the headquarters of the company is. And I'm looking at a beautiful hillside here. It's a great town, about 45 minutes east of San Francisco.
And spring must be upon you now. Weather must be starting to get lovely in California now.
Well, the weather in California is usually pretty lovely. So, we have seasons somewhat, but it's always great. Yeah, I love it here. Came out to California about 26 years ago, and I've loved it here ever since.
Yeah, I've always been an East Coaster since I've moved to the States, but my wife won't let me move to California because I think we'd then never leave, and that's a problem for her. She wants to move around a bit. So
I see. Yeah, it is a little bit of a black hole. It's hard to leave once you're here.
Yeah, 100%. We've got a lot to get into today. There's a lot to go through. I really appreciate you coming on the show. So before we get into the real details of everything, it'd be great to have an intro to yourself and your background, and we can take it from there.
Yeah, sure. So my background is started out as a kind of on the science side of things. I was an applied math major and then got a PhD in biomedical informatics. And coming out of grad school, I was so in grad school, going back to college, kind of had this notion that building companies would be really interesting and really motivational for me. And as much as I was interested in sort of in science and academia, there was sort of a draw toward creating companies in my head. And part of the reason why I was interested in coming out to California in the first place, having read about kind of this is late 90s, early 2000s, about kind of the culture and the allure of the startup world and the technology world out here. And also California, as we just talked about, is just is a beautiful state from what I had read. And when I came out here, it's like found out that everything I read was kind of true. So coming out of grad school, I was on the team that started 23andMe, and my background is in the dry side of the world, so bioinformatics, computational biology, so kind of a mixture of computer science, statistics, and biology. We started 23andMe kind of at this nexus of these, sort of several different trends coming together, where there was these new technologies for genotyping people at scale, where you can actually measure individual, sort of get people's genetics. And then also kind of the advent of what was called back then Web 2.0, meaning kind of dynamic applications on the intranet. And that seemed like a natural marriage, where you can take these like complex data sets and now dynamically display valuable information to people about their genetics. It was a really, really fun experience. We were doing things for the first time that had never been done before. There was like all these amazing papers that were coming out, and you could just kind of take the results in those papers and start kind of relating them to individual people and their individual genetics. We very much saw ourselves as a kind of a dry company, in the sense like a software company, a data and analytics company, where we would build applications and deliver results, but ourselves were not the ones developing the technology to measure genetics. And at some point I found that in a way that in itself is powerful, but it's also constraining, because oftentimes kind of the things that you most want to know about a person's genotype at that time were oftentimes the hardest things to measure. And that kind of put me on this path toward going deeper into the actual tools for measuring biology, and led me onto a path where I joined an early startup that was started by a friend of mine called Quanta Life, where we built technology for measuring biology really precisely using what was called digital PCR. That company was acquired in 2011 by a larger company, Bio-Rad. And, after spending a bit of time there, I, left and, got together with my co-founder, literally a coffee shop in San Francisco. And out of that emerged 10x.
It was interesting that from the top of your introduction, you were entrepreneurial as a motivator early on. It's actually quite rare that we hear that, because mostly we hear great science, love the science. If anything, some people miss the science and they've been thrust into these founder CEO type roles, and at some point try and get back to the science. But you considered yourself motivated by the entrepreneurial spirit quite early on.
Yeah, it's kind of interesting because you look back and you kind of sort through your motivations and your path. And I do think that looking back, I was more motivated by that sort of entrepreneurial bug from fairly early on. It's interesting because I actually remember this particular moment that put me on that path. When I got to college, and that sort of as is maybe typical of immigrant parents, my parents wanted me to be a doctor, and that's what I figured I probably would be. I was interested in biology, and being pre-med was natural. After about 1 year in college, I kind of realized I would make a terrible doctor, and I was just, and there's other reasons where it just didn't appeal to me being a doctor. And I wasn't really sure what I wanted to do. I was really interested in science and a few different sort of big questions in science. But I remember there was a particular point where somewhere midway through college, I went to a panel. And the reason I went to the panel is because the professor that I was working for, whose lab I was working at, was on the panel. And I was kind of curious. And he was both like a very famous academic and also an entrepreneur. And that's why. And the panel was about entrepreneurship. And it had a pretty eclectic set of people on it, right? There was sort of this hyper famous academic who started several biotechs. There was like the founders of a juice company, Nantucket Nectar. Remember, there was a few other people, founders of software startups. There's like a really interesting sort of Q&A. And I remember there was a particular question when someone asked from the audience, is like, well, you guys make it sound so compelling and so interesting, but what about your work-life balance? How do you deal, or something to that effect? How do you deal, like, do you have interest outside of work, and how do you balance those? And one of the panelists, kind of gave an extended answer to that. And at some point, my advisor kind of cut in, and he had that sort of tendency and sort of the kind of maybe in some sense the authority to kind of cut in and just and he said something, as it was his style, something very cutting, which is he pointed to the questioner and told us, like, you know what, the fact that you're asking that question might mean that you're just not fit to be an entrepreneur. And it was, again, a very aggressive kind of thing that, if you're a high-level academic, you tend to be used to saying. Obviously, it wasn't really the kind of thing you oftentimes hear on a panel, but somehow, funny enough, it really appealed to me, this notion of going all in on something and knowing that it's really valuable and committing yourself without reservations to that mission, to that purpose, was really appealing. And so I think, again, I would never, growing up, I grew up in the Soviet Union, and so I never thought about business or entrepreneurship. No one in my family had ever done anything like that. There was none of that. But that moment, I do remember, kind of clicked in my mind, and that's what kind of, from that point on, there was a thread, right? And it's not like I would say at that point I decided absolutely I'm going to be an entrepreneur or whatever, but it did change my mindset. It made me inspired, and I kept coming back through subsequent years. And when in grad school, I kept thinking about, well, you know what, this kind of a thing could be an interesting idea for a company, or some kind of a product, or some kind of value kind of creation thing. And so, yeah, I ended up getting drawn to startups, and ultimately kind of this, again, the notion of building companies was there as a common thread for a long time for me.
That's awesome. And so you were sitting in a coffee shop in San Fran with a friend, and this was the early days of the idea that became 10x. What was it that you guys seen as the problem that existed that you were like, this is what we're going to go solve?
Yeah. Well, so it's kind of interesting, right? These things oftentimes are both sort of a mixture of high level vision and also like a very personal kind of decision. I mean, for me, a lot of it really came down to like where I really like working with Ben. So we were colleagues at our previous company at Quantalife, and Ben is a chemist by training, Ben Heinzen, and he was a chief science officer at Quantalife, and we ended up working really well together. Very different backgrounds scientifically, right? I came from the computational side. He's a chemist, but together we had a really, really productive relationship. It was really, really fun to talk about ideas and see them kind of implemented. I felt, sometimes I would come up with a thought. I would ask him, would this be possible? Not expecting that the answer would be yes. But then, he would kind of look in the distance like, yeah, I think we could do that. And that would just shock me. And, sometimes the reverse or something like the reverse would happen. And that whole relationship, and I would say sort of the experience of Quantalife more broadly, was very, very productive. Again, in contrast to the experience of 23andMe, which was great, but there we were very much saw ourselves as a software company or software genetic company. We hired everyone who was kind of like us. When I got to Quantalife, which was a physical kind of company that was building physical products, no one was initially like me. And it was both very challenging, but also very invigorating. Then that sort of very multidisciplinary milieu of Quantalife was incredibly energizing and productive. So that was sort of one vector that kind of drew me to think about, like a potential starting a company. And Ben and I got together. It was like, at that time we were unemployed after leaving our previous jobs. And some people started reaching out to us as like, hey guys, what are you doing? What are you like? And so I was sat together and was like kind of started sketching out what the world is likely going to look like in the future. Back then, you had sort of this next generation sequencing was starting clearly to take off. This was 2012. And you can sort of extrapolate into the future where, okay, the sequencing capacity is just going to become ubiquitous. And you can build all kinds of applications on top of that. So that's sort of one premise that was on our minds. And then we're also thinking in terms of, okay, what does biology really need? Somewhat was informed by my experience at 23andMe, going back to where, again, like the things that were oftentimes most interesting to measure were the hardest to actually measure. And also there's just fundamental kind of gaps in how we could measure biology. The fundamental unit of biology is a cell. And yet when we measure biology, right, we take, gene expression or other kind of analytes, you take your tissues, you kind of mix the contents of all the cells together, and you measure those mixtures, and you lose all the like critical context. Yeah, there's other applications too. And then as we thought about these different applications, we kind of realized that if you could, a lot of them ultimately reduce to the problem of being able to run many, many reactions in parallel. So whether you're measuring individual molecules or you're measuring individual cells or, immune cell receptors. And if you could have a technology that could create very large numbers of reactions in parallel and marry it with the sequencing capacity that's going to become increasingly prevalent, increasingly powerful, you can build really powerful products that could really kind of advance the future of biology. And so it was sort of this combination of a personal kind of imperative where, hey, like it would be really, really fun and really productive to work with Ben again, and sort of the set of ideas around what kinds of things we could build. And we initially sketched out in a notebook kind of some of the initial approach we could take of kind of scaling the very large number of reactions in biology. And I remember Ben asking, "Is like, hey, maybe we should start a company." I'm like, I think let's do it, right? And it started like kind of that sort of as a really initially as a project, an idea project, and as you kind of proceed through the paces, and we attracted a core group of really, really talented people as a kind of the founding team. You can end up being able to think bigger, more ambitiously, which allows you to bring in even more amazing people. And then kind of sometime during that first year, kind of transition my thinking from being where, hey, this is a really interesting, fun project, interesting company, to where this could be a really, really meaningful company where I can really, going back to my like original motivations, where I could really, together with other people, leave a mark on the world.
And effectively, you guys have then gone on to build a category in your own right. So how has it gone from those initial thoughts of tools to platform to effectively the industry standard of what exists now?
Yeah, it's interesting, right? Like you kind of, there's a time for everything in a sense, and you want to be clear about sort of the initial market point, and you kind of want to focus on the features and your first product and the product market fit of it. And, for us, the story was, we kind of had first several applications mapped out. One of them was long range sequencing. And so that's been still a problem with a lot of next generation sequencing, is that you only get short reads and you don't get the full kind of molecule of DNA, and you have to do a bunch of work to put it back together. So that was one application that we kind of thought was going to be interesting. Single cell analysis, what I talked about earlier, was going to be another, and we had some other ideas. All of them, again, kind of map back to the same sort of foundational platform technology, and that's what we ended up spending our time in the first couple of years really building. And that has been one of the principles that has driven our thinking, and still drives our thinking. Kind of investing in foundations, really important. So you invest a lot up front to make sure you kind of build the right foundation, and that allows you to move a lot faster subsequent to that, to iterate, to build new products, new applications, kind of improve that. And so we build that, and then what ended up becoming the Chromium platform, kind of the foundations for that, the technologies that we invented, the factories that we built to generate the consumable products, the instruments. And our very first product was actually around long range sequencing, and it was a great product. Customers loved it, but unfortunately there wasn't a large enough number of those kinds of customers, as we learned as we got to market. And you always want to be have appropriate sort of epistemic humility before you get to market, because your signals are always going to be, there's always just inherent irreducible uncertainty. And for that reason, partially, before even we got to market with our first product, we started developing our second, which was around single cell analysis, because you just don't know, as much as we were getting great signals, there's just certain uncertainties. And the single cell approach felt to us was orthogonal, was very different, and had a lot of really compelling first principles reasons behind it. And once we got that to market, that definitely hit the really kind of great needs that were there, among researchers, and it took off, right? And so that was a product that really had great product market fit initially, and had great amounts of uptake. And now they're kind of going back to your question, right? You're starting with okay, this tool that really fits the market well, and we're very fortunate to kind of hit upon that. But there you kind of flip into where you now need to really listen to the market and what your customers are saying, and kind of pedal to the metal in terms of iterations, in terms of new applications, in terms of new products. And we were really, really intently focused on delivering all these new products. And so from that first launch, subsequent years, many new analytes, many kind of new advances in terms of just core single cell features and performance that we brought to market. And also, you need to invest in your commercial kind of go-to-market structures to make sure that you can satisfy the demand that's being driven there. And so that kind of, by getting to market with something that was really, really hit the sweet spot of the need, and then iterating and pressing subsequent to that, really allowed us to establish a platform. Really, as we scaled, it sort of became, in many ways, has overtaken sort of maybe the category of tools in general as being kind of the driver of a lot of research now that's happening in the world.
On that, a lot of the tool side has historically stayed in research. What has to happen now to go from sort of discovery into sort of clinical reality?
Yeah, so for us, we've obviously built a lot of products since our initial single cell product, both in terms of building out the single cell capabilities, but also over the past several years launching really powerful spatial biology capabilities, so new platforms, new technologies, new products. But the fact is, up until very recently, a lot of these tools have been very much confined to kind of basic science, basic research. And one reason for that is that it's a natural progress of things. Like when you first bring a new category of measurement, new technology to measure biology, it most naturally gets adoption with scientists who are looking to understand the basics of biology. Now, once a technology is established there, once there's sort of the credibility that comes along with basic science, and it kind of goes through its the customers test it out and validate it, then you can start expanding into more translational applications and ultimately into clinical applications. Now, the thing about single cell in particular that made it maybe more challenging on that front is that it was designed, single cell biology, fundamentally, the technologies that we have had been designed to work with live tissues. And so you're running your experiment, and you take your cells, and while they're still kind of live, you prepare them, you run them through the analysis. Now, that's really powerful and generates lots of amazing discoveries, but makes it very challenging to use it in a translational clinical context, where generally tissues are fixed. They're usually not available. Like, the analysis is very hard to do at the point of sample collection. It's a very prosaic kind of an issue, but it is a really, really fundamental issue. And over the years, we've had many customers who have tried to use these tools in a more translational, more clinical context, but it's very challenging. Certainly, it's very challenging to do it at scale. But over the past several years, we have rolled out capabilities that actually allow you to do both single cell and spatial analysis on fixed tissues, on formalin fixed, paraffin embedded, which is the standard way that tissues are stored in pathology, and on whole blood, like where you can collect whole blood, fix it, and ship it to get analyzed. So that's a relatively recent development, but really sets now the product set on a good foundation for clinical and translational applications. The other thing that's really critical and it's really necessary is generation of large-scale clinical evidence. There's plenty of signal that has been coming out of scientific literature. Now there's over 10,000 papers that have been using our tools that show that there's like really, really important insights you can gain from measuring biology that should affect how you think about treating patients. But there has not been really large scale, like robust clinical studies to really establish that signal. And for that, you need scale. And again, you need products that can be run on clinical samples. And that's what we've also been working on over the past several years. Some of it has to do with reducing costs. Some of it has to do with improving workflows. But now we're at a position where we can really do that. And we're seeing our customers kind of embarking on these large-scale data generation efforts. And also we ourselves are now investing in efforts in specific applications, specifically in cancer and in autoimmunity, to generate clinical evidence. So that's really the next important kind of imperative in order to bring these kinds of technologies into the clinical context. And we feel really, really excited because both from first principles, based on all the empirical data and all the conversations we've been having with our customers, the opportunity here is just massive.
Wow. So the benefits of single cell, the benefits of spatial, ultimately the biggest challenge has been so far, can we bring both of them together at scale? And now 10x are on the verge of being able to do that. Do you want to sort of give us an introduction on what comes next for 10x and the technology and where that's going?
Yeah, we're really excited. Like, this to me is something that has been many years in the making. A new platform that we're launching now called Atera. It is a really big step change and huge step change along multiple dimensions of what is now going to be possible. Kind of the way I see it is sort of maybe in a way the ultimate platform for measuring biology. If you think about kind of what are the key elements, sort of abstraction layers of biology, there's molecules, there's cells, and there's tissues. And historically, we've had different technology stacks, different tools to measure these different areas, and they're all kind of live in their own different markets, and oftentimes different scientists kind of use different tools, right, to separate. So you had molecular biology, which sort of, been culminating in genomics. You had cell biology, you had tissue biology, histology, pathology, and measuring these things, biological systems separately. But really, like, what you need to do is kind of be able to measure it all together at scale, at the right resolution. And Atera really kind of brings it all together for the first time. Spatial biology, that's been the promise of it, that you can measure cells, molecules, and tissues together. But it has been sort of the field has been hampered by a number of constraints. You kind of had all these trade-offs. Like if you want to measure lots of genes, you can only do that really, really low throughput. Or if you try to push the throughput, you can your sort of performance suffers. All kinds of issues. And we've kind of designed Atera from the beginning to kind of really take away all these constraints, where you can now run whole transcriptomes on tissues at this massive throughput, at single cell level sensitivity and specificity. So like really exquisite amount of information, huge amounts of data that you can get from this platform. And, like really, really amazing feedback from just the early previews we've been having with customers, where the feedback is just like, okay, this is like the biggest technology leap they've seen in their careers. And, it is awesome when you're like working internally really hard. The team has been really, really putting their blood, sweat, and tears into this platform for many years now. And when you get to the market and you see this validation coming back at you from customers, it is a really, really like incredible feeling. And I think this is one of those things that happens, looking back at our industry, you see the step changes. And I think Atera is like really one of those step changes that happens maybe once a decade or so, maybe even less than that. And really, really excited to kind of like how it's going to drive the next decade of research and ultimately clinical applications as well.
Incredible, Serge. So what does this mean for the future of disease understanding, biomarker discovery, and diagnostics?
Yeah, well, so the platform itself, is designed to run at really large scale. So in terms of enabling basic discovery, but it also, back to the points I made earlier, we designed it from the beginning to work really well with clinical samples, and to be able to do this at scale. So if you imagine, we certainly expect that's going to drive massive amounts of basic science discovery, but also now thinking about clinical applications. If you have any samples, if you have stored biobanks, or you have patients coming in and you have clinical information about them, and you want to correlate those clinical information, phenotype markers, with the underlying biology, there's going to be no better technology, there's no better approach than running it through Atera. And where this becomes particularly powerful now in the current world of this exponential progress in AI, where you can now actually take very large data sets, very data-rich data sets, and draw insights from them. Atera is perfectly made for this moment. And we do like what we're seeing now already, and what we expect to accelerate going forward in the coming years and then decade and beyond, is this massive acceleration of how we are going to be able to discover new science, and how we're going to translate these discoveries into treatments, and also into precision medicine.
Wow. So we've had early access through certain customers, and they've been giving you feedback. When does a full commercial launch commence, and when do you expect to see that sort of really, going out to the market?
So we're basically starting to take orders now, and the platform is going to be shipping in the second half of this year.
Wow.
And we're starting to take orders also internally before the actual instruments are available, to be able to run samples internally for customers on the platform as well. So yeah, so it's here. It's going to be imminently available. I'm very excited to really go forward and transform how research is done.
I can feel your excitement, Serge. It's and if you're excited, I think everyone should be excited. So it's fascinating to hear about how these elements are all coming together, and at the scale that makes things really exciting in a clinical setting. You've been at you've seen this entire ecosystem sort of being built. I want to sort of ask you a couple of questions about your perspective, because it's fascinating, right? What have you learned in watching this ecosystem be built? What separates great companies from the average ones, and why genomics companies still fail, right? It's not an easy industry to succeed in. So, what do you think from your perspective that makes great companies succeed in this space?
Yeah, well, so I would first of all draw a different kind of a distinction between tools companies like us, like building tools for others to use, and diagnostics companies that use technologies that are available to bring diagnostic tests to market. And there's very different business models with their own sort of complexities and failure modes and success modes. And I can sort of touch on both. And we certainly have ambitions also to kind of turning on diagnostics capabilities in the future. But, if I were to focus on tools specifically, I mean, a lot of it is kind of, it comes down to some extent product market fit initially, right? Are you going to build kind of the right technology? And in particular, what is challenging in the tools market is to build a technology that has a sort of that's big enough to support a sizable organization in a large market. So it really comes down to kind of a market size question. And in a lot of cases, you can have a technology, going back to our initial experience too, that where you can get, the first 50 customers are like relatively easy to get because, there's people who really like technology, right? Scientists who really like technology, and they're always looking for new technologies. But the question is, can you, go beyond that? And that is hard to assess from the beginning. Now, for us, what we try to do is kind of think from first principles. What are the really fundamental, foundational kinds of capabilities that would be general purpose technologies that would enable breakthroughs across a whole range of areas across all of biology. And that's how we go about it. And that's what has allowed us to become successful. If you think about single cell, you think about spatial, you think about all the papers that have been published, there's not an area of biology that I can think of that has not seen like really fundamental discoveries that were driven by tools, right? And so I think that is a really important consideration when you kind of look at successful versus less successful companies. But, if I were to abstract at a level, and that applies beyond just sort of tools, in many cases, I think just obsession with talent, quite honestly, is like, I would almost say it's sort of a necessary and sufficient condition, both, right? Like everything is downstream of talent. And I think if I were to trace our experience and our success to one thing, I would say is that from day one, we wanted to work with the very best people. We wanted to surround ourselves with the top experts in the world, whether it was, particular technical areas, whether it was business, whether it was other areas. And when I look at companies out there that are successful, that tends to be very much the common ingredient, is that sort of obsession with talent.
Do you think that obsession with talent goes hand in hand with the ability to stay focused on innovation? In ultimately, when a company becomes commercialized and a commercial engine, there's a lot of pressure to continue that engine growth, right? Do you think that the obsession with exceptional talent, who typically are motivated by innovation themselves as an individual, do you think that allows you to stay focused on new innovation?
Yeah, I mean, that's an interesting question. I think it's part of it, for sure. But I do want to say that talent doesn't mean just talent in R&D, talent in creating sort of new products. I kind of, when I think about talent, I think about it across the whole company, whether
Right.
it's your finance department, whether it's your commercial organization, whether it's your G&A functions, all of that, or their operations. And I think, certainly innovation is a big part, and for us, that's kind of the core of what we do. And in fact, we're a pretty big outlier, I would say, relative to other companies, given our scale, how much we invest in R&D and how much we invest in innovation. It does, you kind of bring up the point, like as you scale, it does become harder because you have financial pressures, and oftentimes the easiest way to kind of respond to those is to kind of shrink back innovation. I think that's a mistake. Fundamentally, long-term value creation comes, certainly for a company like ours, from innovating and for bringing new capabilities to market. And I think that's ultimately, that has been a source of our success. It's going to be a source of our success and source of outsized value creation. But I also want to kind of stress that it doesn't have to be, it's hard, but it doesn't have to be an either or. Like, we are focused right now very much on driving really great financial performance as a company. And if you have great finance people, you've got great business people, great commercial people, yes, you can do that. And in fact, it becomes a core part of you fulfilling your mission. Like, our fundamental mission is around advancing science and advancing human health. It's to accelerate the mastery of biology to advance human health. I see that like financial performance as being part and parcel and very closely intertwined with the mission, because the only way we can accomplish our mission is by virtue of having resources to feed the innovation engine, right? To feed R&D, to build new capabilities. And the only way we can do that is we're like really successful financially. It is a harder sort of task, maybe, to be able to do both simultaneously, but it is the one that, while harder, is also the one that creates absolute disproportionate value in the long run. And I think our experience so far has been evidence of that.
So, based on that, what's the What's the 10x culture like? And what are the types of people who really thrive in your culture?
Yeah. So, from the beginning, when we started the company, we had that very multidisciplinary core like partially by design, because that's where Ben and I came from, that experience, and we're really motivated by that. And partially by being fortunate, kind of in the initial founding team, of attracting really, really great people from different disciplines. And so when I think about people who thrive here, first of all, just like people who really are really curious and are eager to learn from others who are really experts in other fields. And so I think that entails being both sort of having intellectual curiosity, having intellectual horsepower to like think through things that are outside of your comfort zone, and also like a measure of humility, right? Being able to work with others who are experts and kind of appreciating their expertise, which oftentimes doesn't, it's not necessarily like a super natural human sort of emotion, because if you spend decades becoming an expert at a particular thing, you would tend to think that that thing is the most valuable thing that exists, right?
Sure.
And it's somewhat unnatural to kind of to take that back and like make yourself kind of vulnerable and learn from others. And I think that's really, really important. People who are fundamentally like real hard workers and driven by impact, right? Like who want to create like awesome things in the world. Back to your question, who thrives here? Like I think that's a really, really important consideration and an important driver.
That makes sense. Does that make it, because a lot of companies that really scale but also continue to innovate, they almost proudly become places where this isn't for everyone. We're trying to change the world here, and that means that there's a really high bar, that sort of top 1% mentality. Does that mean you have to have that sort of emphasis on this isn't for everyone, and intentionally make it a difficult place to sort of to really sort of maintain that?
Yeah, look, I think going back to my my personal motivations, I like being kind of all in on working on something that's really, really important, where I have
Right.
no qualms about me putting in my time and working with others and kind of pulling other people along and creating something like of tremendous value in the world. And given our mission, what we're trying to create is kind of definitionally is really challenging. Like we're always working on creating things that have not existed in the world before, right? Kind of pushing the frontiers of science, frontiers of what's possible, and with technology, what's possible with business. And so work by definition has to be hard. And I think it's really important to be upfront with people. Back to the question of like who would thrive here, who is not. Like you want to be clear that yes, like we're working on hard problems that requires a lot of commitment. It requires really hard work, and you want to be clear with people upfront that that's the kind of place we are. And it does tend to select for people who are of like mind, and kind of fosters the right culture for that reason. And so, yeah, like for us, we're always looking for those, kind of category defining people who are really eager to make an impact in the world. Lots of intellectual horsepower, lots of drive, and kind of a measure of humility.
Yeah, I love that. I always find it's the companies that go out and do special things that have that same relentless standard on this. There's a really high bar to get in here, but also to survive here. They just go on and achieve, productivity wise is just that multiple. Where do you see over the next decade the skills that are really going to matter the most in the industry?
Yeah, like I mean, this is the big question for everyone in the world right now, right? It's really interesting. I don't know if I have necessarily a better answer than lots of other people who are thinking through this right now. The big change, of course, is AI, and it's changing how we work. And I think it's going to change like fundamentally, kind of the whole idea of what an organization is, what a company is, and my view is it's going to be really, really important for people to be able to use these tools, these technologies to the max. I think the people who can use them the best, who are most adept in this world of these of AI tools, agents, are going to be the ones who are going to thrive, right? And we're all trying to figure out like what does that mean exactly, and it definitely means at the very least like working at a higher level of abstraction than many of us have you have been used to, you're going to have really, really powerful capabilities at your beck and call. The question is how you are able to use them. And in some sense, everyone will have to become a manager, right? And like a manager of organizations in a sense of AIs and AI agents. So I think kind of being most adept in this new world is probably the most critical, most important thing, which kind of pushes forward on critical thinking and will push people to operate at a higher level of abstraction than they've done in the past.
Yeah, I like that. And I think it also goes back to what you said about that curiosity, right? I think people have to be more curious than ever right now. If you're sitting in your job that you've done for a long time and you're not willing to learn new things, there is something coming very quickly right now that you're being chased almost, and that's a real danger. That curiosity is going to become very obvious over the next few years, the people who have put themselves out there to learn new things and stay ahead of things.
Yeah, no, absolutely. I mean, there's so much happening continuously. There's so much to learn, for sure. The curious mind here is absolutely going to be like a huge advantage to folks.
And so you're at the forefront of what will fundamentally change in biology over the next sort of 5 to 10 years, especially with the launch of Atera. Where do you see the industry going from here, and what are you most excited about?
Well, yeah. So I think there's sort of three things that have been on my mind, kind of like when you think at like really, abstract up, like what are the big high level trends? So first of all, like, of course, AI, like, and huge progress being made. And I mean, there's a lot of investment, a lot of activity in deploying AI tools to biology. And it's happening kind of in two places. One is with agentic science, kind of closing the data analysis gap. Where historically, especially with the sort of genomics, high kind of throughput technologies, you've been able to generate data, but then how do you go from data to insight, right? To actual understanding. And now there's this amazing progress that's been happening with agentic AI scientists to be able to kind of close the gap, and I think it's gonna continue, and it's really just going to accelerate science in a huge way. For the last 2-3 decades, we've been kind of stuck in this world where we've been just limited by the number of bioinformatics people that are available to analyze data, and all of a sudden that bottleneck is getting addressed. It's really exciting. The other thing about AI is that is going to be used to represent biology in increasingly powerful ways, right? To make these kind of predictors of biology, virtual models of cells, tissues, molecules. This is happening. This is just the beginning. There's good reasons to kind of think that there's lots and lots of scaling ahead, and as you scale the amount of data and the size of these models, they'll become more and more powerful. And to scale them, you need to generate lots and lots of data. It's very similar to what's been happening in other industries where AI has had a huge impact. The difference, at least compared to LLMs, for LLMs there was the internet that they could be trained on. In biology, there is no internet for these models to train on, and so we need to generate huge amounts of data. And it's really going to kind of change the paradigm of science in many ways, where the models themselves will be the drivers of more data generation, as opposed to being sort of the thing you apply at the end to do your data analysis. And now, complementary to sort of that progress in AI is progress in technologies to measure biology. We've designed, to some extent, 10x for this moment. We built it back in the day. Kind of our premise is we need to accelerate science. We need to accelerate the understanding of biology. To do that, you need to measure biology the right way. Biology is really complex, which means you need to measure high resolution at massive scale. That was the premise, right, for all our kind of initial ideas and products, and what's been driving us since. Now, what AI needs precisely in this moment is large-scale generation of the right kinds of data at high resolution, large scale. And for us, of course, again, the issue has always been, as you generate more and more data, how do you translate that data into impact? And AI is a perfect complement to that. So I think these two kind of revolutions, one driven by, in many ways, Atera, and the other one by progress in AI, are going to be mutually accelerating over the next 5-10 years. And then as you think about sort of the impact of these technologies, where I think you're going to see like acceleration of the basic understanding of science, but then also kind of translation of that in the clinical realm. And I think where people what we're seeing, and I think there's sort of people often, I think, are maybe missing just kind of these big kind of things that are changing, trends. We're getting to a point where it's getting easier and easier to create drugs for different targets. We have a growing set of modalities. We have increasing efficiency with how we can, if we have a target, how to affect it. But what's becoming the crux of the challenge is, when do we give it, like, what is the right clinical context to deploy a particular drug or drug combination? How do you match the medicine to the right patient, the right disease? And that's sort of the thesis statement, the problem statement for precision medicine. And so as we're increasing, as the number of potential drugs, potential modalities is increasing, there's increasing need for diagnostics, but for measuring biology to guide treatment, to guide those medicines. And at the same time, as there's the increasing need for diagnostics, for measuring biology, we're getting increasingly good at measuring biology, right? And we're driving that revolution. And so as we look forward to the next 5-10 years, there's this confluence of trends that's really going to put precision medicine, the whole concept of precision medicine, on an exponential curve. And the value of diagnostics is just going to explode. And we're certainly looking forward to doing our part in driving that revolution.
Serge, you got me excited. I think obviously your perspective is so well respected, and to hear how you see these things coming together for how it's going to impact patients' lives in the reality, people that don't necessarily understand the science or don't have the time to keep up with it, what they're going to experience in precision medicine is so exciting. But we're not just talking fluff. What you're presenting here today is the reality of these things coming together and how quickly things are going to change. That's incredible to hear. So I really appreciate you coming on and obviously sharing the news about Atera. Incredible, so exciting to see how things develop over the next sort of 12 months with that commercial launch. But your perspective on the industry, where we're going and how we're going to get there is so valued. I appreciate your time so much.
Well, thank you. It was a lot of fun. I really appreciate the conversation. Thank you, David.
Thank you, Serge.